Hi guys, welcome to Year 10 Science, Genetics and Biotechnology. And the next one in our series is DNA replication. In the last video, we had a look at the two processes of cell division, which is mitosis and meiosis. Now this involves a copying of the genetic material in order to produce cells that are either identical for growth or repair, or to produce cells that have half the number of chromosomes of the parent. And these ones are going to be used as the sex cells, the ones that are going to carry the transmission of genes into the next generation. These two processes of mitosis and meiosis require an exact copying of the DNA. The material must be passed from one generation to the next successfully in order for those traits that have been successful in one generation to make it through to the next one. And this is the raw material upon which evolution operates. The copying of the DNA is called DNA replication. This is quite a complex process with a number of different chemicals involved, and we won't need to go to it in a lot of detail at this stage of your course. Of course, there's a lot of information on the internet, so if you wish to have a look at some of that to expand your knowledge in this area, feel free to do so. One of the most important things to remember when we're looking at the replication of DNA is the complementarity of the bases. And what that means is that when we look at the process of DNA replication, we're looking at consistency of linking of our George Clooney's, our guanine and cytosines, with our Tony Abbott's, our thymine and adenines. So these pairings are known as complements, and they are the basis upon which DNA replication occurs. In addition, we need to look at the concept of nucleotides. Now, one of the things that happens during mitosis and meiosis is the nuclear membrane breaks down. That is, the very thin membrane that surrounds the nucleus and holds all of the genetic material within breaks down, which means that the components of the cytoplasm, all of the chemicals that are present within the cell, can actually come into contact with some of the contents of the nucleus previously separated by that membrane. And that means free nucleotides, and a nucleotide is a sugar phosphate base unit. And as we mentioned before, the base could be one of G, C, T, or A. And this sugar phosphate base unit is called a nucleotide. Free nucleotides are necessary within the nucleus in order for us to bond to the growing chain of DNA. So here's a diagrammatic representation, and whilst I won't go through this in a lot of detail, there are a couple of important things to be aware of. Firstly, you can see that what's basically happening here is we have something that is kind of like a zipper. An enzyme, DNA polymerase, is an enzyme, okay, which is just a type of protein. And we've mentioned the fact that we have enzymes, for example, in our saliva that help us to break down starches. Enzymes are chemicals that catalyze reactions, that is, they regulate the speed. Generally speaking, they will increase the speed of a reaction and facilitate some reaction to happen that may not spontaneously happen on its own. So what we want is an enzyme to come in and cut the DNA strand apart. As it does this, the DNA strand starts to unzip like a zipper, exposing those bases. So you can see here is the section of bases. This section along here is where our sugars and phosphates would be. And so we have the exposed bases here looking for their complement. The free nucleotides over here, which now, because they're in the cytoplasm and the nuclear membrane is broken down, they're now able to get in contact with the DNA strand. You can see there's a section of the DNA which we call the leading strand. That strand is being copied as we go along. So you can see the new strand already starting to form at the top. And then there's what's called a lagging strand, which is the other side of the original DNA, which is moving again with the help of an enzyme, in this case called DNA polymerase. And the DNA polymerase is the one that produces this very long, complicated DNA molecule. The helicase is another type of enzyme, and you can see it's the one that's actually driving through the section of DNA that is forcing it to push apart like a zipper. The bases are going to continue to fill in under these rules of Tony Abbott and George Clooney in order to make sure that we have the exact copy of the DNA that we had originally. So what you can see is that one strand of DNA here is becoming two identical strands over where the enzymes are doing their work. So just then to quickly summarize that process, we have enzymes, special proteins, which cut the DNA between the bases. The strands separate like a zipper, exposing the bases. Spare nucleotides that are in the nucleus they bond chemically to the exposed bases of the nucleotides that are part of the DNA strands, or the two halves of the DNA. And as a consequence, we get two complete and, of course, identical strands that are going to form. Just a couple of little terms that you might come across when you look at this process in a little bit more detail. I haven't gone through any of the names of the enzymes. It's not important for you to remember them at this stage, but you certainly can have a look at those if you want. And you'll notice that enzymes are characterized by and large by the ASE ending to them, such as amylase in your saliva. So firstly, we'll look at the fact that when the chromosomes are copied, so the process of mitosis and meiosis starts with that DNA replication process, and that means that the chromosomes will double. And that doubling of the chromosomes means that we pair them off and attach them together at a point called the centromere. It's at this point where you have a doubling of the chromosomes that when you look at them down a microscope, they tend to look like an X shape. Now, chromosomes most of the time don't look like that, but then most of the time we can't see them. They're so coiled up and concentrated that they're too small for us to see with our microscopes. But during the processes of cell division, the doubling and the thickening helps us to see them, to make them look a little bit like an X. And that's one of the reasons why you will often see chromosomes represented by the X. Each strand of those doubled chromosomes is called a chromatid. So two of those together, joined along the centromere, is your double chromosome. Before replication, each single chromosome represented one molecule of DNA, one sequence of bases with sugars and phosphates holding them together, representing one copy of your genetic information. But after replication, the chromosomes have doubled. So now we have two molecules of DNA representing the two complete copies of your DNA, those two chromatids, as I said, joined at the centromere. So here's a question for you. Why do you think the DNA needs to be copied exactly? We'll have a look at that in class. Thank you for watching.